Hello students, this is SriRam Shastri. I once again welcome you back to the new video session. So today we are going to discuss about the cleavage and in the next topic we'll discuss about the implantation. So what is cleavage? Cleavage is nothing but the division of the zygote after the fertilization for the formation of the embryo. So the zygote is a structure which is diploid after fertilization. The haploid set of chromosomes of the sperm and haploid set of chromosomes of the ovum, they fuse and they form a diploid nucleus called as the zygotic nucleus, and the fertilized egg is now called as zygote. So you can see the diagram here. Okay, so this zygote undergoes division. It is similar to mitosis but not exactly like mitosis. It is equational division where in the number of chromosomes divided in the cell division of zygotic division is also equal, but it is not very exactly same like that of the mitosis. What are the major differences if you talk about? Each cell here after the division of the zygote is called as blastomere. Each blastomere is responsibly can be developed into a separate organism. Okay, so they are totipotent in nature. So embryonic cells are totipotent. That means each single blastomere can develop into a separate individual with the same genetic composition. Clear? So this is the concept of twins, how twins are formed. Already in the last video sessions I have already discussed about how are twins formed and different types of twins. You can refer the last videos. Okay, so the first zygotic division occurs just within 24 to 30 hours of the fertilization. So within 30 hours of fertilization, the first division of zygote starts, and the first division is from a longitudinal axis, that is from animal pole and vegetal pole. Okay, you can see the diagram. Okay, so it is holoblastic and equal cleavage. It is holoblastic and equal cleavage. The cells formed here are equal because they are not laden with egg. Human ovum is not containing huge amount of yolk. It is very less. It is microlecital or alecital egg, we can say. So here what is happening, the division is longitudinal, it is holoblastic and equal. It forms two cells. From two there will be horizontal cleavage and it results in 4-cell stage. So 2-cell stage, and then its division results into 4-cell stage, then 8 to 16 cells. Now this ball of 16 cells, what you call, it's called as morula. The meaning of the word morula is the structure similarly looks like that of a mulberry. Mulberry, if you know, Morus alba is the scientific name of mulberry. So this plant is been cultivated generally for sericulture because the leaves of these plants are consumed by the caterpillars of the what you call a silk moth. The caterpillars of the silk moth feed on these leaves, and in sericulture the plant called mulberry is being cultivated. It is a plant which also develops fruits. These fruits are small purple colored what you call as lustrous fruit. Okay, it looks very similar to that of blackberry, but it's not. Okay, they are having small berries, a bunch of berries forming a composite fruit like structure. Okay, so here what is happening here is that this fruit has a resemblance with a group of cells of the embryonic development, so we call it as morula. Morula looks similar to mulberry's fruit. So all 16 cells are totipotent. After that there will be formation of blastocyst. Now in human beings the blastula is called as blastocyst. Why blastula is called as blastocyst is, it looks, if you see the structure here, the blastocyst looks as if it is been covered by a cyst. The zona pellucida exists. Inside that what happens? There is a trophoectoderm or trophoblast layer. You can see the diagram very clearly here. Okay, so this is trophoectoderm or also called as the trophoblast layer, this cell layer what I am talking about. Okay, so these cells are present and they form the outer covering like structure, and there is a inner mass of cells. So it looks like encysted structure, and that is the reason why it is called as blastocyst. Okay, so human blastula is called as blastocyst. give the reason. This type of questions are asked. Okay, so blastocyst, because it looks like a cyst in which mass of cells are enclosed, right? After this stage, the gastrulation takes place, and then there will be implantation. Then in the next video we'll discuss about how does the implantation take place. Clear? So here the what you call as the cavity is present. This cavity is called as blastocele. This is the inner mass of cell, and this is outer zona pellucida layer. Okay. So up to this, it's cleavage. Once cleavage is completed, then gastrulation starts. At the stage of gastrulation in blastocyst, generally blastocyst, when it is formed, immediately at that stage there will be implantation. Implantation is a process where the embryo gets embedded inside the uterine tissue, and there will be formation of chorion and extra embryonic membranes for the nourishment and development of the fetus. In the next video, we'll discuss about the details of implantation. So you have to remember a few things here, like the first cleavage takes place in 30 hours, then 60 hours, second cleavage 72 hours, in third cleavage, and so on and so forth, like that. Okay, I hope you have understood what type of cleavage takes place in human beings and what is cleavage and its a process. So we'll stop this video here itself. In the next video session, we'll continue with the process of implantation. Thank you.